Hello, and welcome to the latest Technology Networks webinar, Deciphering Cell-to-Cell Interactions in Liver Metastases. I'm your moderator, Alexander Beadle, science writer for Technology Networks, and I'm excited to be here to host today's session. We have a fantastic presenter, Dr. Mathie Lavidon, who will be sharing her valuable insights with us. Dr. Lavidon is a postdoc researcher in the laboratory of Professor Martin Gilliams at the Flanders Institute for Biotechnology Centre for Inflammation Research in Belgium. She obtained her PhD in 2021 at the Sorbonne University in Paris, where she studied the modulation of macrophage niches during tumour development and in response to chemotherapy. After the presentation, we will have a Q&A session, and we encourage you to submit your questions at any time during the presentation. To do so, simply type your question into the box on the right-hand side of your screen and click Send. We will do our best to address as many questions as possible within the time we have available. If you encounter any technical difficulties during the webinar, please click the chat box on the right-hand side of your screen to request assistance from our support team. Now, without further ado, I will hand over to our speaker, Dr. Lavidon.
Good afternoon, everyone. First, I'd like to thank the organizer as well as Miltenyi for giving me the opportunity to present my work, which focuses on the cells and interactions driving the immune response in the metastatic liver. So our lab has been interested in characterizing the signals controlling macrophage development, maintenance, and function, which we define as the macrophage niche, with a particular interest for the liver. The resident macrophage population of the liver is called Kupffer cells, and our lab has shown that the identity of Kupffer cells is programmed through signals delivered by neighboring cells from the niche, namely endothelial cells, stellate cells, and hepatocytes. Of course, to be able to identify the different signals at play in the different niches, the first step is to know what are tissues. So with this idea, and to have a wider understanding of the liver environment, and in a collaborative effort with Charlotte Scott's lab, we've generated an atlas of the liver encompassing cell content, gene and protein expression, and the spatial arrangement using mRNA and protein information for each cell type. We performed site sequencing on the whole liver to gain information on both mRNA and protein expression and determine specific markers for all our favorite liver populations. And so combining RNA and protein allowed us to have better clustering and also better definition of the population and identify markers for this population, either the mRNA or protein level. And so to spatially identify the distribution of this population, classical microscopy was not really an option, as you become quickly limited by the number of markers that you can use. And so we therefore turned to the Maxima platform in collaboration with Miltenyi, and we used a 100-plex protein panel through cycles of staining and bleaching. And using this technology, we could now spatially define the different immune subset of the liver at single cell resolution. So now, for example, we were able to study like Casey distribution, as you can see here, like really lining with the sinusoids. But we were also able to have information of like the endothelium, like the network with like portal vein and central vein, and like all of the other immune subset distributions, so T cells, B cell, monocytes, and also fibroblasts, and all in one section. So this just gave us a lot of information, and this was really key to define like our atlas at the spatial level, and to really be able to resolve the definition of the niche by defining cell-cell interactions. We were also able to visualize the zonation pattern at the protein level. So it is known that in the liver there is a zonation, so a gradient from portal vein to central vein, where you have different endothelial subset and hepatocytes, because they have different metabolic function of the liver. So this is based on like the processing of the different nutrients. And here you can really see it clearly here, where you have a different type of endothelial cell here in the portal vein. And then as you move towards the central vein, you have like different markers that are expressed around like all those sinusoids and also the vein itself. And what was really cool is that we also found back all those structure in human samples. So this is healthy liver, human liver. And so we can find again like all the immune cell distribution, so all the Kupffer cells, but also like the portal vein, the central vein, the cholangiocytes, like all those structures that we were able to identify in only one sample. And so for now, I'm only going to show you the steady state that we did for the liver atlas paper. But if you're interested, this is all published work, and we really made a massive work to define like every like structural cell and macrophage and like immune subset that you can find in the liver. So just a piece of advice when you design your mix experiment, so your maxima panel, it is a good idea to pre-test your antibodies using conventional microscopy, just because it's just going to make your panel design easier for the future. Now for my project, I focus more specifically on colon cancer-derived liver metastases. And we know that metastases are difficult to detect early, which make them even harder to treat. And it's been shown that like cancer dissemination to the liver does not only impact our liver immunity, but systemic immunity, which makes it even harder to treat and also very low responsive to immunotherapy. So there's really a crucial need to understand what happens in the like metastatic liver environment. In addition, we also know that there are two distinct growth patterns that are found in patients. So the desmoplastic that is characterized by this dense fibrotic rim that is like, we don't really know what it is, but we think it's like some fibroblasts and maybe like dead hepatocytes or something that are kind of crushed together, but they really separate the tumor tissue from the healthy tissue. And then you have the replacement pattern, which mimics more the liver architecture, where like it all kind of comes together and you have lower immune infiltrates. And it's been shown that the replacement growth pattern correlates with worse prognosis than the desmoplastic one. So it would also be key for us to understand like what drives one pattern or the other and what's actually involved in the structure of those growth patterns. So if we go back to the BRIC, like hypothesis, I'm interested in understanding how they are reorganized and maybe even reshaped during liver metastasis. So on this picture, you have the metastatic nodule here, and it's surrounded by a capsule of fibroblasts, and the healthy liver is all around. So as we have deep expertise in Kupffer cell identity, I was first interested in understanding what was happening to the macrophage niche, like the Kupffer cell niche, upon metastatic development. And so to address, and what is striking on that image, so here is that in pink you have the Kupffer cells, and in green you have monocyte-derived macrophages. And so you can see that the Kupffer cell here, they are excluded from the metastatic lesion. So they do not infiltrate the tumor. And you can only see them in the healthy tissue and at the border, but never, like, very rarely in the nodule. However, we do have a population of macrophages infiltrated here, which are monocyte-derived. And in addition, so there is like some kind of signals that prevents the Kupffer cells from infiltrating the metastasis. And in addition to the macrophage subsets, I also analyzed stellate cells. So those are the resident fibroblasts of the liver parenchyma. So they are in red here. And I also stained for like general fibroblasts, so in yellow. And so I'm going to show you a zoom here because it's clearer. But what I observed is that in the healthy tissue here, you find your stellate cells in the parenchyma as expected, so they're red. But then if you look within the nodules, you can see fibroblasts that are only yellow. So meaning those do not derive from stellate cells, and they are like recruited or like derived from another structure and arrive in the nodule. On the other hand, though, at the border here, between the metastasis and the healthy tissue, you can see that, again, you have red and yellow fibroblasts, indicating that those ones are derived from stellate cells that were present in the tissue before metastasis development, but they acquire a new morphology at the contact of cancer cells. So it's kind of like they were hijacked by, or like co-opted by the tumor to support its growth. So you can see like it's kind of like an activated phenotype that is really different from the stellate you can find in the healthy tissue. I also performed staining for endothelial cells to try and identify subsets specific to the metastasis. The same way that we hypothesized stellate cells are co-opted by the cancer cells, endothelial cells could also be used by the metastatic nodule to support its development. So at the moment, we do not have reliable marker to distinguish blood vessels that were present in the healthy tissue and could have been co-opted versus blood vessels that would be newly formed, but this is a hypothesis that we have. And so altogether, the hypothesis is that like those observations suggest that the metastatic growth induces the formation of distinct macrophage niche. So for example, here you would have kind of like the normal KC with like normal stellate cells and LSEG, but that start to interact with the cancer cells. And then you would have one population of monocyte-derived macrophages that interact with the co-opted stellate cells and maybe the endothelial cells. And then here, monocyte-derived macrophages that interact with fibroblasts that are not derived from stellate cells with maybe a different type of endothelial cells. So we could imagine that there is a co-option of stellate cells, for example, by the cancer cells, and they could be responsible for delivering new signals to KC at the tumor border. We were interested in like validating if our observation made in the mouse liver were also like translating to human health, like human samples. So we were interested in the modification of macrophage niche in metastatic human livers. So we applied the Maxima technology to paraffin-embedded human metastatic liver, thanks to a collaboration we have with Peter Vermeulen in Antwerp. And we used this time a 200-plex tumor microenvironment panel to determine the different liver cells that are in the metastasis. By looking at it quickly here with like about 10 markers, you can already tell that there are different tissue architectures, cell distribution, and cell interactions in the different zones of the liver. So here you have the healthy part of the tissue, and then you have the tumor site. So for example, in red, you can see that Kupffer cells are only found in the healthy part of the tissue, which is also what we observed in the mouse model. And at the border of the tumor, they acquire a new morphology. So this is nice that our mouse model is able to recapitulate partially at least what happens in humans. And then here you have like what we think are monocyte-derived macrophages because they do not harbor any Kupffer cells markers. And then we also like identify different type of fibroblasts that could constitute those different macrophage niche maybe. But this is only 10 markers and we have like 200. So how do you analyze that? So the hypothesis again is that here we have those different niche like that are spatially different in the metastatic liver. For the analysis, I started by segmenting the cells in a region of interest that encompass both tumor and healthy zone, just because it's faster to process than taking the whole sample at this point. And this is done with the MaxIQView software from Miltenyi. So you have quite a lot of tools that are available directly from the software. So then I performed dimensional reduction and cell clustering just on a few populations to already see if we could discriminate a pattern. And what was very interesting is that, as I said before, like there are no KC within the metastatic nodule, and they really seem to be retained at the border. Whereas within the nodule, we observed what we assume to be monocyte-derived macrophages. And then, so in the tumor, you can also see like you have like infiltration of like T cell and dendritic cells. And so I wondered if there was maybe a correlation between the distribution of some macrophage population and T cells. And then to address this question, I generated some correlation metrics to determine the colocalization between the CD8 T cell in cases or tumor-associated macrophages. So what you can see here is that overall in the tissue, the correlation between KC and CD8 T cells is not high. However, we do have clusters at the border of both population colocalizing. So this could indicate that the activation status of KC is different in this part of the tissue and in that part of the tissue. And maybe this could partially explain how the metastasis is going to progress. And now if we focus on CD8 T cells and TAMs, We observed some tumor zones enriched for CD8 T cells, colocalizing with tumor-associated macrophages. And what was also interesting, which is not shown on that image, is that the tumor-associated macrophages in close vicinity to CD8 T cells express higher level of HLA-DR compared to the ones that are not in contact. So maybe there are some like macrophage subset that are more involved in antigen presentation, and depending on how they're distributed in the tumor, this could like give information on the kind of growth pattern that we find, and maybe we could correlate that with like the immune activation in those different zones. And so to be able to address this, we also wanted to characterize like those different growth patterns. So with all the markers that we have now, do we see any like specific organization or like specific population of maybe fibroblasts, because we hypothesized there quite a lot of fibroblasts. This was done on a PFS sample this time, just because we wanted to compare like the FFP and the PFA samples. And so we analyzed different regions of the human metastatic liver. And so both of these are the central region of two different patients. And like I would say that the overall architecture is quite similar. So you have fibroblast accumulation and collagen deposition all around the tumor. But on the other, if you look at the periphery, here it's quite striking that you have a different organization. So you can see in the desmoplastic pattern, you have way more collagen deposition and myosin accumulation, which would suggest indeed that you have like more fibroblast activation and accumulation. And so maybe you have this kind of like fibrous capsule that separates the cancer cells from the tumor, and like in a way it kind of isolates the tumor and prevents it from damaging more the healthy tissue, whereas in the replacement you see like it's more all together, and like the cancer is really gonna invade like in between the hepatocytes and everything, and you don't really have this containment of the cancer. So for us, having the ability to screen these samples with 200 markers is an incredible tool, and ideally would like to combine it with automated analysis to determine the common features that we find between patients exhibiting a same growth pattern. So as I mentioned, we need automated analysis with this kind of data set because it is a lot of data, which make it quite hard to analyze manually. And we want to, as much as possible, to have unsupervised analysis to avoid any bias. So this is what a PhD student from our lab, Michiel, is working on. So he started zonation markers to try and determine the portal to central vein axis on all the liver, just to like train the algorithm. And then, so once he had like those kind of axes determined, he analyzed Kc, like he mapped Kc distribution along this axis, which revealed a gradient of Kc from portal to central vein. Now this worked well when we looked at one cell population using like 3 markers, but one of the main issues of automated analysis is cell segmentation and how we can split our cell in a highly heterogeneous tissue with cells closely interacting with each other. So in collaboration with Evans-Sai's lab, we've been working on how cell segmentation can be optimized, especially from mRNA imaging. And so why are we so interested in mRNA? It's because for some markers, it's very hard to have only the protein. So for some cell population, you're not going to have an antibody that's good, like STAIN for a membrane marker, but also a lot of activation markers, such as cytokine or like some secreted factors. It's going to be hard to track which cell produces it. So then having the ability to stain at the mRNA level, it's very good. But then the problem is that your cell segmentation become harder because, for example, here we've stained our 4 bricks of the casein each, and we've tried to determine to which cell belong each mRNA. And if you look at the initial segmentation, we have a cell that contains RNA of endothelial cell, hepatocytes, and Kupffer cells, which is very unlikely. And now, because we know those cells are entangled with each other, what's more likely to happen is that this is not actually the shape of a cell, but this is what you would expect. Like this would be your Kupffer cell, just because, as you can see here by microscopy, it's really like all together and it's like elongated cell. So it's not just one flat cell. They have like protrusion that kind of go all over the place. So in the end, Kupffer cells would look more like this and this. And this is why we believe that the combination of RNA and protein is going to be necessary to have the capacity to define the cell boundaries, but also the activation state of all the cells of interest. And that's why very recently we have been beta testers for the RNAsky technology from Miltenyi that allows us to combine imaging of mRNA and protein, which will be an incredible tool to resolve single-cell spatial organization. And it's going to give her more information about the activation state of the cells. So just to show you the preliminary results that we got, this is the steady-state liver of the mice, where we could identify our favorite population at the RNA level, also by combining protein markers for the blood vessels. So we have the Kupffer cells, the endothelial cells, the stellate cells, and the cholangiocytes. And because we know those populations, they really like exhibit the patterns that we would expect from those cells. So this was really exciting. And then we've also tried it on human samples of liver metastases to get better characterization of the two growth patterns. So we took like from one sample those two growth patterns with a 67 protein panel plus 6 additional antibodies and a 24 RNA custom design panel for the RNAsky results that we got. So this is combining mRNA and protein for a different fibroblast population. And after gating the different population, identify distinct distribution pattern for fibroblasts, macrophages, and T cells, depending on the growth pattern. So these are really preliminary results, and we're now working on getting more samples and deeper analysis for this data. But it's already really exciting, and we really think this is going to be the next step for us to be able to identify like the specific activation profile of fibroblasts and macrophages, which we think is going to be key interaction to like define the type of growth pattern that we're going to get and how this is going to play for a prognosis associated to this growth pattern. So with that, I want to thank everybody involved in this different project. The Liver Atlas has really been a massive collaboration effort between our team and Charlie's team. I've also had a lot of help from the microscopy core, from Miltenyi, and from our lab for the Maxima experiment. So I really want to thank everyone involved, and I thank you for your attention. And if you have any question, I would be happy to take them. Thank you.
Thank you for your attention, everyone. And thank you, Dr. Laviron, for your insightful presentation today. We will now move on to the Q&A session. Please feel free to submit your questions using the box on the right-hand side of your screen and click Send. We'll do our best to address as many questions as time allows today. Now, we've already received several questions during the course of the webinar presentation, and so I'll start with those now. So the first question I have is, what type of immune infiltrate do you expect for each growth pattern?
Hey, thank you for the question. So actually, this is one of the questions that we want to address, but we do know that in the desmoplastic growth pattern, we have more immune infiltrates, and the hypothesis is that that's why it correlates with better prognosis. And in the replacement, we expect more like immunosuppressive population, and so we really want to characterize like the kind of circuits that's going to drive the different immune recruitment in both growth patterns.
Fantastic, thank you. The second question I have for you here is, what is the profile of the fibroblasts in the different niche?
Yeah, so this is like also something we need more data on it, but we know that there are way more fibroblasts. Like in the desmoplastic, you really have like this fibrotic ring around the tumor that's kind of like protecting or like isolating it from the rest of the tissue. And what we saw with the Maxima data on the human samples is that you have like fibroblasts that are activated in both cases, but indeed they don't secrete the same kind of protein. So what we think is that you have like more like fibrosis in the desmoplastic ring, and this is what like prevents the tumor to grow too much compared to the replacement growth pattern, and maybe that's why you have better prognosis with the desmoplastic growth pattern.
That's great, thank you. Another question that's just come in here. Is there any information on time points for metastatic growth, or metastasic growth, sorry, or point of entry of the tumor cells?
So we work on colon cancer liver metastases. So the point of entry is it's through the portal vein. So like the vein that you have between the spleen and the liver. This is what we know, like from the model we use at least. So I don't know if you mean also like for the human samples or if it's for the mouse samples, because we did both. For the mouse samples, depending on the tumor line that we use, it's about 2 to 3 weeks to grow. For human samples, we don't really have information about that because it's resection. Sometimes it's done before chemotherapy, sometimes after. Sometimes it's just as a biopsy, or sometimes it's really like for resection. So this is really variable. So I don't have a lot of information on that.
No, that's a great answer. Thank you. Another question here that's just come in. May I ask, what method did you use to probe RNA in FFPE tissues in situ? Also, what markers do you use to define cancer cells?
So if the question is with the RNAsky, which I guess it is, it's the new like technology that Miltenyi just developed, and that allows you to combine the RNA and the protein staining. So you first have a step of like cycles of staining for the RNA molecule. So it's like fluorescent probes, just like fluorescence in situ hybridization. But then because it's like cycle of imaging, you can do like we did up to 27, I think. And then we combine this with the protein, so the Maxima technology that already we worked with before. So it's all on the same machine, and it's like all like one like protocol, let's say, where you can combine the RNA and the protein. And to define the cancer cells, so here we it's mostly like the cytokeratin that we used, because we know it's like overexpressed in the cancer cells of colon cancer derived liver metastases. And so by combining different cytokeratin that were present in the immuno-oncology panel from the Maxima platform, we were able to identify your cancer cells.
Thanks very much. The following question we received actually was, how do you combine protein and RNA? And I suppose we've addressed that already, but I don't know if you have anything else that you'd like to add.
Yeah, so here, like from the data I showed, it's indeed the RNAsky, like so with the combination of the RNA and protein, it's with the Maxima platform, like the Maxima machine using the RNA technology. So yeah.
Fantastic. The next question that I have for you here, can you easily determine to which cell each RNA molecule belongs to?
Yeah, so this is what we're working on now. It's actually like knowing, especially when you have a cell that's not like round, how do you know to which RNA molecule it belongs? And this is why we were really excited to be able to combine it with the protein, because then you get like the cell membrane, like the border of your cells. So this is really like crucial for us. Because otherwise we did in the past where we didn't have the protein signal, and we have to like get rid of some RNA molecule when we're not sure which cell it belongs to, because we tend to like focus on the nucleus then to be sure that it's really in that cell. So the combination of both really allows us to have like more RNA molecule attributed to a cell, which is really like really interesting for a lot of markers that are actually more found in the cytoplasm than in the nucleus.
That's a great answer. Thank you so much. the next question that I have here, how do you suggest to design your antibody panel?
So the way we did, so we've tried the immuno-oncology panel from Maxima, but then we've also, for the liver atlas paper, what we had is the combination of the single cell and the site-seq data. So we had a lot of information about the transcriptomic, but also the proteomics of the cells. And the site-seq really allowed us to screen for a lot of antibodies that then we tried by confocal, because then we really had information for all our cells of interest, and then how we could spatially determine them using the maxima.
Thank you. Another question here. What do you think about the co-expression of RNA and protein? Could you always see that if you have the RNA signal, you could also detect the protein signal?
So I guess it's for a same marker in that question. So because the RNA protocols are a bit harsh, sometimes we do lose some protein expression. But we did some validation that for example, for Kupffer cells, we're able to recover Physic4 RNA and protein co-expression, or like for F. So that was really nice and clean. So we were able, for example, to highlight the interaction between Kupffer cells and stellate cells. But you want to keep in mind that indeed, because of the protocol that your sample is going to go through, you might lose some protein staining for some antigen that are a bit more delicate. But yeah, that's really how we started, with just a few markers to confirm that the expression of the RNA and the protein level was really found in the same cell.
That's great, thank you. Another one here. A question from an audience member who'd like to know if you use DFT simulations in your work at all.
I don't know what is DFT simulation, so I guess I didn't use it. Or maybe I did use it, but I just call it something else. I'm sorry, I don't know what is DFT simulation.
No problem at all. Unfortunately, we are coming close to the end of the time that we have available today, so I'll just pick one final question here to wrap up. And we'll go with this one. How do you like working with the Maxima, and what is your highlight of using the platform?
I think, yeah, it's quite user-friendly, I would say, because it all comes in a kit, and the protocol is really quite clear and well explained. So it does require a bit of optimization, because yeah, it's going to depend on your sample and what kind of population you want to detect. So you might need different incubation time, or to try different antibodies. But if you have a panel that's set up, the actual experimental part is quite straightforward. So yeah, that was a good experience. I guess then it's still a lot of work with the analysis, because we get a lot of data from it. So we are excited to go deeper into it.
That's great. Thank you very much.
Thank you.
I'm afraid that is all the time we have for questions today, but please remember that any questions we did not get to will be answered offline as soon as possible. You can also continue to ask questions even whilst watching this webinar on demand. Don't forget to download your certificate of attendance, which is available from the Handouts tab on the right-hand side of the webinar platform. Once again, we appreciate your time and participation today. Thank you, everyone.